Hello students, we are on the last part of this fifth chapter, cell structure and organization. By far in this chapter we have studied about most of the cell organelles. We studied about the cell wall, the cell membrane, where we studied about the fluid mosaic model given by Singer and Nicholson. Then the cytoplasm, cytoskeleton, nucleus, Golgi apparatus and lysosomes. Now in this particular session we are going to study the remaining cell organelles and we are going to start with endoplasmic reticulum. Now endoplasmic reticulum, they are of two types. Either it can be a rough endoplasmic reticulum and the smooth endoplasmic reticulum. The rough endoplasmic reticulum, they contain ribosomes, and the smooth endoplasmic reticulum, they lack ribosome, and that is the reason that they appear smooth. So if you see in this picture over here, this is the rough endoplasmic reticulum where you will see the ribosomes attached to the outer surface, and this is the smooth endoplasmic reticulum, that is without the ribosomes. So endoplasmic reticulum, they are little networks within the cytosols which are present in all eukaryotic cells except for the ova and the mature RBC. So you have to remember this, that rough endoplasmic reticulum, it is not present in two types of cells. First, an ova, and second, a mature RBC, a mature red blood corpuscle. Other than these two, all the eukaryotic cells contain endoplasmic reticulum, and all of them contain both the rough and the smooth endoplasmic reticulum. Now, rough endoplasmic reticulum, they form 50% of the total membrane of the eukaryotic cell, and the smooth endoplasmic reticulum, it is involved in various processes in different cells. So the function of the smooth endoplasmic reticulum, it differs from cell to cell. Now depending upon the cell type, it helps in synthesis of lipid. So there are, for example, you can take steroid secreting cells of the cortical region of adrenal gland, testis and the ovaries. Then there is detoxification of drugs and poison. So these are the two examples for smooth endoplasmic reticulum. Rough endoplasmic reticulum, they are primarily involved in protein synthesis. Okay, so these proteins, they are secreted by the ribosomes that are attached to this rough endoplasmic reticulum. Okay, and so they are called as secretory proteins. So the pancreatic cells that secrete insulin is something that you can take an example for the rough endoplasmic reticulum. So this is about rough endoplasmic reticulum. The next cell organelle is mitochondria. Now mitochondria, these are very important cell organelles as they are involved in aerobic respiration. And there is one thing about mitochondria, that it is absent in prokaryotic cells. That means it will be absent in bacterial cells, and it is also absent in mature RBC. Okay, so in these two things, mitochondria is completely absent. Now mitochondria, as you can see here in the picture, it is a double membrane bound organelle, and the shape of it can be oval, it can be spherical, or it can be a strip like, it can be a spiral strip like. Now they are known as I said, they are involved in aerobic respiration. They are called as powerhouse of the cell. They are the ones that are responsible for producing energy. Okay, so they are known as the powerhouse of the cell. Now this double membranous structure, in this the outer membrane, it is permeable to various metabolites due to a certain presence of protein, that is a porin, or you can call it as Parson particle. Okay, so these proteins, or that is the porin and the Parson particles, they help some of the material to get in the mitochondria. Okay, so that is how the outer membrane is structured, it becomes permeable. Inner membrane is selectively permeable. Okay, so it is selectively permeable to few substance, and both are separated by a space outer chamber. Okay, you have to remember this again. The outer membrane of the mitochondria, it is completely permeable to various metabolites due to the presence of protein. and these proteins help the metabolite to get inside the mitochondria or go out of the mitochondria. And the inner membrane, it is selectively permeable only for few substances. It is not completely permeable. Now the inner membrane, if you speak about only the inner membrane, it is divided into folds. So if you see, these are the folds which are known as crista. Okay, and they bear numerous particles which are known as oxysomes and cytochromes, or they are also called as electron carrier. So the inner membrane, it encloses a cavity, and this cavity contains a fluid matrix. Now the matrix that is there enclosed inside the mitochondria or the inner membrane or the mitochondria, they contain few coils of circular DNA. There are, as you can see over here, then there are RNAs. There is a 70S type of ribosome. So ribosome are of two types that we'll be studying in the next slide. For mitochondria, the mitochondria has 70S type of ribosomes. There are certain lipids and various enzymes which are involved in Krebs cycle and the other pathways. Okay, so this was about the mitochondria. Now let us also speak about oxysomes that are attached to the crista of the mitochondria. These oxysomes, as you can see the picture over here, now if you see over here, the small dotted structures that you can see, these are the cristas. Okay, these are the oxysomes which are attached to the crista. Now let us study about the oxysomes. So one thing, that they are attached to the inner membrane of the mitochondria, and they are either known as F1, F0 particle, that is Fernandez-Moron particle, elementary particles, or mitochondrial particles. Each particle, it will have this head structure, and it will have a stalk and the foot. Okay, the head is the F1 particle and the foot is F0 particle. The head, it faces towards the matrix. So if you see, this is the crista, this is one fold. Okay, and on this fold you will see the oxysomes are attached, where you will see that the head, it faces towards the matrix that is enclosed in the inner membrane of the mitochondria, whereas the foot, you will see that it is embedded in this inner membrane. Okay, so the head, it acts as an enzyme ATP synthase, and the foot are the proton channels, and altogether oxysomes, they are involved in proton pumping and ATP synthesis, and that is how mitochondria becomes the powerhouse. As it produces energy, it does the transaction of energy by synthesis of ATP. It is considered to be the powerhouse of cell. Then the next organelle are plastids. Now plastids are again double membrane structures, and they are found only in plant cells. So plastids, they are absent in animal cells, they are only present in plant cells. Plastids are of three types: chloroplast, chromoplast, and leucoplast. Chloroplast, they contain chlorophyll, that is the green pigment which is found in plants, which is responsible for the green coloration of the plant, and they do the function of absorption of sunlight, which is necessary, and it is a raw material, it is a source of energy for the process of photosynthesis. Okay, so chloroplast, what are chloroplast? Chloroplast are the structures which contain chlorophyll. What are chlorophyll? Chlorophylls are the green pigments which are present in plants, and these pigments, they are responsible for the absorption of sunlight, which is required for the process of photosynthesis. Other than this, they produce and they store glucose as a stored food. It is a food storage organ, you can say. Then comes the chromoplast. Chromoplast, it contains the pigment carotenoid, which is responsible for the red, orange, or yellow pigment, the red, orange, and yellow coloration, and it is mainly found in flowers and fruits. Okay, so the flowers and the fruits that appear red, orange, or yellow in color, it is because of this pigment carotenoid which is enclosed in chromoplast. And then the third type of plastid is leucoplast, which contains no pigment. Okay, and it is used to store starch. Okay, so these are the three types of plastid: chloroplast, chromoplast, and leucoplast. And it is only present in plant, it is not present in animal cells. The next cell organelle is vacuoles. Now vacuoles, they are membrane bound sac which are prominently found in plant cells. In animal cells, if we speak about animal cells, wherever and whenever it is present, they are present in a very minimum amount, few in number, and they are smaller in size compared to the plant cells. The vacuoles which are present in animal cell, they are, first of all, they are very few in numbers, and if at all they are present, they are very small in size. Now generally there are 2 or 3 permanent vacuoles in plant cells. In some of the plant cells, the large when we take an example of a large plant cell, a single large vacuole is present that occupies the central part of the cell. Okay, and it is considered to be a central vacuole. Now this type of vacuole, it occupies as much as 90% of the total volume of that particular plant cell. Okay, now the vacuoles, they are bound by a semi-permeable membrane which is known as tonoplast membrane. Okay, it is a semi-permeable membrane. That means this membrane, it will not permit anything and everything inside the vacuole. That it is limited to certain metabolites and certain particles only. Now this membrane, it helps in maintaining the composition of the vacuolar fluid, the cell sap, and it keeps it different from the cytosol. Now the composition of the cell sap, it again differs from cell to cell. Okay, now the cell sap of the central vacuole that we just spoke about over here, it is a storehouse of various ions, and thus it is hypertonic to cytosol. And small vacuoles in seeds of certain plants, they store organic materials like proteins. Now other than this, in vacuoles, along with excretory product, other compounds are also stored that are harmful and they are unpalatable to herbivores. Now when we say they are unpalatable to herbivores and it is present in the plant cell, that means these vacuoles and these excretory products, the stored compounds, they protect the plants from these herbivores so that the herbivores don't come and graze upon these plants. Other than this, if we see the attractive colors of the petals of the flowers of the plant, they are also due to such storage pigments which are present in these vacuoles. Okay, so vacuoles, along with the excretory product, they contain other compounds also. First, that there are harmful and unpalatable material which are unpalatable for herbivores, so that they can protect the plant from the herbivores. And other than this, there are certain stored pigments, which are responsible for attractive colors in the petals of the flower. Now intake of the food or foreign particle by phagocytosis also involves the formation of food vacuole. So when we say phagocytosis, as you can see over here in this picture, endocytosis and exocytosis, they are the fundamental 2 processes of intracellular digestion. So the food particles that are taken up by the cells, as you can see over here, the food particles which are taken up by the cells, they get into the cell via endocytosis. So what is endocytosis? It is the food particles entering into the cell. Okay, and after they enter into the cell, they release certain digestive enzymes to extract nutrients, and the leftover waste product of the digestion, it is carried to the plasma membrane by the vacuoles, and they are eliminated through the process of exocytosis. So if you see over here how they are eliminated, the lysosomes, they come and they get attached to the vacuole, and they release all the digestive enzyme of the extract nutrients, and then the waste material is eliminated by the process of exocytosis. Okay, So intake of food or foreign particles by phagocytosis involves this formation of food vacuoles. These are known as food vacuoles. Now in freshwater unicellular forms like paramecium, excretion and osmoregulation also takes place by contractile vacuoles. So with this, one more thing, that vacuoles, it is helpful in maintaining the turgidity of the cell. So students, this was about the vacuole. Now let us get on to glyoxysomes. Glyoxysomes, they are membrane bound organelles that contain enzymes which are responsible for converting fatty acid to sugar. So glyoxysomes, they are observed in cells of germinating seeds, where the cells will utilize the stored fats as a source of sugar till it starts photosynthesizing on its own. Ribosomes. Ribosomes are the particles which are, you know, you all are very much aware of the ribosomes, that there are the ribosomes, they are protein factories of the cell. Okay, just now when we were studying the endoplasmic reticulum, we studied that the ribosomes which are present on the rough endoplasmic reticulum, they are protein synthesizing particles. Okay, so we know that, we are aware that the ribosomes, they are protein factories for the cells, and as we say that they are protein factories of the cells, they use the genetic information to synthesize these proteins. Now ribosomes, they were first observed by a scientist whose name was Pallid in the year 1953, and he observed the ribosomes as a dense particle in the electron micrograph of a cell. Now ribosomes, as said, that they are made up of ribosomal RNA and they are made up of protein, and they do not have any membranous covering around them. Okay, in eukaryotic cells, ribosomes are present in mitochondria, it is present in plastids, and it is present in cytosol in a free form also. So ribosomes, they are either found attached on the surface of this rough endoplasmic reticulum and the nuclear membrane, or they are just freely suspended in the cytoplasm of the cell. Now speaking about the type of ribosomes, as I said previously also, that there are two types of ribosomes. Prokaryotic ribosome is called 70S ribosome, and eukaryotic ribosome is called 80S ribosome. Okay, now the ribosomes, they are made up of two subunits. So in 70S ribosome, that is the prokaryotic ribosome, the larger subunit is a 50S subunit and the smaller one is a 30S subunit. And in eukaryotic ribosome, which is an 80S ribosome, the larger subunit is 60S and the smaller one is 40S. Okay, so the ribosomes are of two types. The bound ribosomes, they generally produce protein. What do you mean by the bound ribosomes? The ribosomes which are bound on the rough endoplasmic reticulum. Okay, they are the ones which are generally doing the function of protein synthesis and are transported outside the cell after the processing in endoplasmic reticulum and the Golgi body. The example of bound ribosomes are acinar cells of pancreas, which produce pancreatic digestive enzymes. And if we speak about free ribosomes, the freely suspended ribosomes, the free ribosomes come together to form chains of polyribosomes of protein synthesis. Now let us study about the last part of this chapter, that is cilia and flagella. Now cilia and flagella, they are fine hair-like membrane-bound protoplasmic outgrowth that occurs on the free surface of the cell. Now these are responsible for generating the current in the fluid medium for the passage of the material and for the locomotion process. Now if we compare the cilia and the flagella, cilia, they are smaller in size and they are more in number. Flagella, they are longer and the cilia, they are smaller in size but they are many in number, and flagella, they are longer but they are few in number. Cilia, it acts as an oar causing the movement of the cell. Now the flagella, these are present in prokaryotic bacteria, and they are structurally different from that of the eukaryotic flagella. The cilium and the flagellum, they consist of the basal body, the basal plate and the shaft. Okay, and it is derived from the centriole. It has 9 peripheral triplets of the fibrils. So if you see this, these are the 9 peripheral triplets of the fibrils, and the shaft, it is exposed part of the cilia or the flagella. It consists of two parts, the sheath and the oxonium. So this is the oxonium, and above this oxonium there is a sheath. The sheath, it is a covering membrane of the cilium or the flagellum. Okay, now the core is called oxonium, which possesses 11 fibrils. Now if we say 11 fibrils, so these 11 fibrils, as you can see over here, they are running parallel to this long axis. It shows 9 peripherals, so these are 9 peripheral doublets, and then there are 2 single central fibrils. So that is how 9 plus 2, it becomes 11. Okay, so there are 11 fibrils running parallel into this long axis. Now this sheath is connected to one of the tubule of the peripheral doublets by a radial spoke. So here you can see that there are radial spokes which are present, and the central tubule, it is connected to each other by bridges. The peripheral doublets are connected to each other through a linkers or the inter-doublet bridge. Okay, so this is about cilia and flagella. Now we also spoke about the centriole in this part. Centrioles and centromeres. First of all, centromeres, they are usually found near the nucleus of an animal cell, and it contains a pair of cylindrical structures which are called as centrioles. The cylinders are perpendicular to each other, and they are surrounded by an amorphous substance which is known as pericentriolar material. Okay, so if you see it here, this is a centrosome which is present near the nucleus. Now each cylinder of a centriole, it is made up of 9 sets of triplets. So this is considered to be one triplet, and like that there are 9 sets of these triplets, and these triplets are called as microtubules. These microtubules, they are made up of tubulin. Now they are evenly spaced triplets and are connected to each other by a means of a non-tubulin protein. At the proximal end of the centriole, there is a set of tubule called hub. So this is a set of tubule which is called hub, which is here present at the center. The peripheral triplets, they are connected to the hub by the means of these radial spokes. So these are the radial spokes that are running between each of these sets of triplets. Okay, so this is the hub, these are the triplets, and in between, the connection between the triplet and the hub is this radial spoke. So that centrosome, it helps in the assembly of the spindle apparatus. During the process of cell division, it forms the basal body of the cilia and the flagella. So if you see over here, the basal body of the cilia and the flagella, it is made up by the centrosome. So students, with this we end this chapter over here. In the next session, we'll be starting with the 6th lesson, that is biomolecules.